Hi, good morning all. Sorry for this confusion of 11:30 and 11 o'clock and 11:30. Actually, earlier the classes used to be 9:30 to 10:30, 10:30 to 11:30, and so on. So I had that timing in mind actually. And still there is a half an hour gap between 11 to 12 and 12. Then you have class from 12:30 onwards. So I thought I can utilize that 30 minutes window. So I hope some of you have managed to go through those videos that I posted. And as I did not see any queries or any questions posted on the group, I believe you guys are fine with those lectures. I mean, I won't be pushing or emphasizing too much on those because they are introductory lectures, just to give you a fair idea, especially the people coming from the non-biotechnology background, a fair idea of what we actually mean by plant biotechnology and what will be the course of movement or course of path for the course. Yes, Yokendra, you're trying to say something.
Sir, Hitesh here, sir.
Huh.
I have not watched all those videos. So, like, if we, I've started just, I have watched first and second one.
Hmm.
So, after completing, if we, I have any query or I will ask, sir.
Okay, that's fine actually. That's fine actually. So without delaying it any further, I will start with the first topic of ours, which is the plant tissue culture. All right. So I'll start with this topic, and I hope everybody has access to the course structure. And as you can see, the first topic starts with the plant tissue culture, and then we talk about the different aspects of plant tissue culture, the different types of tissue culture that we use, and what are the benefits and advantages, and we move on actually. So, have any one of you, or I mean, do you guys have any experience with plant tissue culture? Have you studied this before? Have
Yes,
you
sir. Practically done.
you have done
Yes,
that practical? You have done the practicals also.
Yes, sir.
yes, sir.
So you are quite well aware. I mean, quite well aware of the topic. Like you have studied before.
Yes, sir. Yes.
Yes, sir.
All right, that's good actually. So I don't have to go into too much detail, but I'll try to, because it's part of the course structure. So we'll move as the in alignment with the course structure. So those of you know is quite good enough, but those who are not who who have not gone through, or who are not who are not accustomed with this, it is specifically for those. So I have taken some pictures of plantlets growing in the tissue culture conditions. And I mean, we do here tomato tissue culture here in the lab, because I use the tissue culture method, tomato tissue culture method, for generating transgenics. And this is just to provide a glimpse. We'll go on with the definitions and stuffs in the later slides. But this is just to provide a glimpse that this is how we take a explant, and we allow it to germinate. Then we allow it to form a callus, and this is how, using tissue culture, a specific, a proper medium that allows it for rooting and shooting, and you start getting the plantlets. And then we take plantlets out from here and move them to individual jars, media jars, where we allow them to grow, and then we lead it to hardening. And I mean, that will lead to the development of full plant actually. So this is where, I mean, it's a brief definition, an introduction to plant tissue culture. Plant tissue culture is actually defined as culturing plant seeds, organs, explants, tissues, or cells of protoplast on a chemically defined synthetic nutrient media. I mean, this is very, very important, because a lot of research has already been done in the field of plant tissue culture. I'll take you through a brief history of the PTC as well. So that clearly shows that a lot of research has already been done. And I mean, you might find it interesting that is still one of the very important and very significant field of study, a field of research, plant tissue culture. I mean, of course, they are not sticking to the basic plant tissue culture now. It is being used for more application purposes, or more uh, uh, rather than moving on from basic research to more application oriented research. But PTC is still quite commonly used in many labs, and is still a very interesting area of research, a very significant area of research. So it is defined as culturing plant seeds, organs, explants, tissues, cells, or protoplast on a chemically defined synthetic nutrient media under sterile and controlled condition of light, temperature, and humidity, obviously. And it includes several variations such as meristem culture for propagation of virus-free plants, protoplast culture, cell suspension culture, tissue or organ culture, and anthropoid culture. We'll look at these different variations briefly in the later slides. And I have put one photo of Gottlieb Haberland. He is considered as the father of plant tissue culture. And I mean, this excerpt that I have taken here is actually from one of his papers that he has presented in a conference way back in 1902. And what I want you to focus on, or what I want to emphasize here, is that almost more than 120 years ago, this guy has actually, like at the time he started, he actually had this vision that why PTC, like that plant cells have this concept of totipotency. I hope you guys know what totipotency is. Yes.
Yes,
Yes,
sir.
sir.
Yeah. Please keep responding. Actually, yeah. Please keep responding. So he visualized or envisaged at that time that the plants have this this cellular totipotency, and how the cellular totipotency can be exploited to the benefit of like improving the plant as a whole. In terms of, I mean, there are, you will see later that the plants are being used as bioreactors and all. So, I mean, he has actually envisioned all these things way back in more than 120 years ago. So this is from the paper he presented. So he said that usually a cell is an elementary organ as well as an elementary organism. And it is not only a part of a living unit, but it is a living unit itself. And hence each cell can multiply on its own, actually. And so he was able to maintain such living cells, individual cells, in a living condition using a suitable nutrient media for several weeks. And they later on died after 3 to 4 weeks. But the individual cells that he extracted from different plant species, from the leaves of the different plant species, he managed to maintain them on a suitable nutrient media for about 3 to 4 weeks. So he actually cultured the single cells in different plant species in a simple nutrient media called NOT solution. And then what he wanted to achieve, he wanted to develop actually full plantlets or plants from these individually growing cells, although he did not succeed at that time. Please mute the audio. Whosoever is it, please mute the audio. So he could not succeed at that time, but his idea was to develop plantlet from individual plants by cultivating them or by growing them onto a suitable nutrient media. And that was his vision at that time, actually. But he could not succeed. But of course, he gave the impetus to start considering PTC as a area of active research. And before him, we had the Sheldon-Sezwan cell theory. And he actually was working to prove that, I mean, that's where he started. He was actually trying to prove the cell theory correct. So based on the cell theory where individual cell is a living unit and can survive on its own. So he wanted to verify the concept of cellular totipotency proposed by the cell theory. And that's how he started. And his objective was to get a full plantlet by culturing a single cell. And he proposed that the cells in the plant body, they stop growing after acquiring the required features, but they do not lose the inherent potentiality to keep growing further whenever they meet a suitable nutrient solution. And because he also put forward that embryos can be obtained, until then it was the concept that embryos can only be obtained from the gametic cells, from the androecium and gynoecium, but he proposed that the embryos can also be obtained from vegetative cells. And because of his vision, or because of his the idea that he had, he is considered as the father of plant tissue culture. And these are, I have not listed all of them, but these are some of the major landmark discoveries that form the part of the history of plant tissue culture. Of course, it's a long list. If you want, I can share with you guys, but I've just tried to pull out the very, very significant ones during the course of the plant tissue culture history. So there was a guy called Liebeck. He managed to develop hybrid embryos using nutrient media by taking, he was generating a cross, and whatever seeds were being formed by the cross of these two species, they were actually aborting prematurely. So he was not getting plants. So he decided to use this tissue culture method for obtaining full hybrid plants by taking out the embryo after the crossing and culturing it on a nutrient media. So this actually paved the way for embryo culture. Now, this is where the embryo culture actually started. Then in 1934, there was another guy called P.R. White. He developed modified synthetic media by mixing inorganic salts, yeast extract, and sugar. And he showed that the media is sufficient for continuous growth of roots of tomato, for continuously growing root cultures of tomato. And then later he replaced the yeast extract with 3B vitamin B, pyridoxine, thiamine, and nicotinic acids. So this actually showed the importance of having vitamins in the plant tissue culture media. And if you have done some plant tissue culture experiments, as you said, have you prepared plant tissue culture media? Hello.
Yes, sir. We have. Yeah, we have.
So what all components did you include? Like what all did you add?
Sir, basically first agar for support, then the hormones which are required, and our teacher also instructed to put us some antifungal agents. I don't remember the names. And kanamycin. Yes, sir.
That's it.
Sorry, not antibacterial agents.
Yeah, not antifungal, antibacterial agent, kanamycin.
Yeah, I don't remember the name of exact what was the antibacterial and antifungal agent, but we did put some. We did put some. So avoid we were actually producing callus, sir.
Okay. From which plant?
Moong seeds, moong explants, sir.
Which where did you do the experiment? Which lab? Like where did you bachelor's?
From Mumbai University, sir.
Mumbai University. Okay.
Yes.
So what did you not put? I mean, you named agar, you named hormones. Yeah, and you named some antibacterial agents, antibacterial antibiotics.
Sir, major salts, minor salts, vitamins.
Yeah, salts and sucrose as
yes,
well.
exactly.
Some sugar as well.
Yeah, you missed the
I
major,
forgot to mention that.
macronutrients and the micronutrients, the major salts and the minor
I forgot
salts.
to mention that.
Yeah. That's what I was trying to get at actually. And then did you not include any vitamin solution?
We didn't. Yes, we didn't. We put yeast extract.
That's all right actually, but it varies from plant to plant, from species to species, and of course it also depends on what kind of experimental requirements actually. Like whatever experimental requirements you have, it also depends on that. But when we do it, we add vitamin B solutions. We add vitamin B stocks in there, actually vitamin stock in there. So what I'm trying to get at actually that it is an outcome of the research done almost like 90 years ago, almost 90 or 85 years ago, where they found that vitamins are a crucial ingredient of the nutrient solution that we provide to the plants. And we still use that information. We still use vitamin solutions as one of the components when we prepare tissue culture media. Okay, so that's what I was trying to get in. Of course, the macronutrients and the micronutrients you mentioned, the sugar, the sucrose we add, so that that takes care of the sugars, and the inorganic salts is all the macro and micronutrients. So, and do not forget that we are talking about 85 years ago, 90 years ago. So this was still the thing in progress. It was still the work in progress. I mean, people were trying different things to get to a full-fledged self-sustainable nutrient solution. So they were trying. So, but P.R. White is remembered for actually providing the basis or the baseline for one of the suitable media of its kind that can be utilized for tissue culture purpose. Then 1946, scientist called Ball, he succeeded in raising whole plants from shoot tip. And we have different, so this is how it goes on. Actually, I'm not going to read all of it, but you guys can go through. But what I want to focus on is, have you heard of MS Medium? Like all those of you guys who have done plant tissue culture experiments, have you heard of MS Medium?
Yes, sir.
Yes, sir.
What does that stand for? Like, what is it?
Murashige and Coog.
Murashige
again is Coog. Yeah, that's right.
Coog, yeah.
So, I mean, it was prepared, or it was, how to say, it was synthesized in 1962 by two scientists named Murashige and Coog. And the important thing is that it was such based on all the research that was done prior to that, it has been so extensively thought over and extensively tested and prepared that it is still considered as the gold standard for tissue culture experiments even today. Like even after 40-50 years, even in our lab or in most of the plant tissue culture labs that we use, we do plant tissue culture, we use the MS Medium actually. So it is such a gold standard for all the tissue culture, plant tissue culture experiments that we do. And I would also like to highlight here, and I feel proud in highlighting that I am actually a distant lineage. I follow, like I'm a part of a lineage that connects me to Skoog actually, because Skoog, there was a student named Trevor who did his PhD under Skoog. My PhD supervisor did his PhD under Trevor. And of course, I did my PhD under my PhD supervisor. So it's a long lineage, but I feel proud in sharing this actually. Anyways, then coming on to to the Indian perspective of tissue culture, it was actually started in 1957 in the Department of Botany, Delhi University, under the leadership of Dr. Pachanan Maheshwari. And then there were several big names, one of the big names you might have heard about, Guha and Maheshwari. Have you heard of Guha and Maheshwari?
Yes, sir.
Yes, sir.
Yeah, so you've been through all these things before, so I'll not spend too much time on this. So I mean, one of the major discoveries for which Guha and Maheshwari are famous is that they did the anther culture from Dhatura. And they led to the formation of like haploid plants using anther culture. And of course, I don't know if you have heard of Bhojwani also, SS Bhojwani.
Yes, sir.
Okay, good. So
No, sir.
no, okay. Those of you don't know, he is still, I mean, he is still there. He is in the Dayal Bagh Educational Institute in Agra. And he is one of the pillars of Indian plant tissue culture arena. So he's still there. He's quite old actually. And there are several, he has written several books on plant tissue culture, and that is quite widely accepted in India and abroad. Even the slides that, I mean, I am also referring to one of his books, Bhojwani and Dante. So, I mean, and it's a very, very well written book, very descriptive, very vivid. And of course, he's quite famous in the Indian plant tissue culture arena. He's very old now. I've met him once in one of the conferences in 2019. So he's quite old, but he's still there, is still working actually. I mean, mainly into administration and all. So, I mean, this is part of the, like, just to introduce you guys to some of the Indian plant tissue culture experts. And this was I mean, it was in 1956 actually. So it's not following the trend, I mean, the sequence. But I wanted to highlight this here because that was the first time when a company, a Japanese company, Mitsui Petrochemical, used plant tissue culture. They got the what do you call? They got the patent for this. So to synthesize chiconin from using plant tissue culture. So they used cell culture media. They used cell cultures in bioreactors, and for the synthesis of chiconin. So this was the, I mean, they got the patent for this actually. So, and they started the large scale culture of plant cells in a bioreactor. So it's all that old actually. And these are the major areas of plant tissue culture, the different variations of plant tissue culture that I was referring to earlier. So you have the intact meristem culture, you have the explants, different types of explants, and you have the cell suspension culture that can also be initiated from callus cultures. Then you have the protoplast culture and anther microspore culture, and they all can lead to plants by different pathways or different ways. And I mean, they all have their specific advantages or their specific requirements. Like protoplast culture, anther culture is more used in plant breeding methods, more required in plant breeding for plant breeding purposes. And then the cell suspension culture, as I mentioned before, this production of secondary metabolites. So the chiconin is a secondary metabolite. Ginseng and Taxol are also being produced in bulk. You're using plant cell culture. So this cell suspension culture is mainly used for secondary metabolite production. And then meristem culture and callus formation for plant propagation and plant breeding. And so moving on, I will go through these in a very brief manner individually. So first, the callus culture. Now, callus is an irregular mass of parenchymatous tissues, and it lacks any organized structure, any organized growth, but it keeps on dividing. So it shows tremendous cellular differentiation. This is what a callus looks like if you guys have seen it before, or I don't know. So this is what a callus looks like. So you have some explants and they start get into de-differentiation. They have already been differentiated into leaf or stem part or root, whatever. They have already been differentiated into different organs. But then when you put on a suitable media as a combination of auxin and cytokinin, they start showing de-differentiation, and due to tremendous cellular differentiation, they form callus-like structure, which looks like this actually. I mean, talking about callus, so we use this method, the callus culture method, very routinely in the lab, because this is what we use for generating transgenic tissue culture experiments leading to transgenic preparation. That is plant propagation, what I was talking here actually. So depending upon the crop, depending upon the plant that we are targeting, the explant will vary actually. The explant will vary. Like in tomato, we use cotyledonary leaves as the explant. In rice, we use the dehusked grain as the explant. And so it varies in chickpea, we use the embryo, we remove the cotyledons, we use the embryo as the explant. So it varies from plant to plant. I mean, this has been an outcome of the research that is being done in the previous decade or so. But this varies from plant to plant actually. And you need to keep on a favorable media. The explant starts showing cell division, and it initially starts at the edges. We see all these stages like when we do the tomato transformation here. the tomato tissue culture, and then it actually ends up covering the entire explant thing. And then after 3 to 4 weeks, like we keep doing subculturing. After every 15 or 20 days, we keep doing subculturing. So from these callus, you will start seeing, based on the media that we are putting it on, you will start seeing some kind of differentiation. You will start seeing some root formation happening or some shoot formation happening. So we take that part out and we put them onto individual sterile glass jar, and we allow that plantlet to form roots and start forming a plantlet, which can later be under aseptic conditions, which can later be allowed to grow into a bigger plantlet, and then we transfer it to soil eventually. Now, this is the second form of tissue culture, the cell suspension culture, second variation called the cell suspension culture. With reference to the callus culture, where one cell is actually communicating with another cell via plasmodesmata. But in a cell suspension culture, ideally, all the cells, you have individual cells and each cell starts multiplying. But this is an ideal condition. I mean, we not every time or not always we get individual cells culturing. You might have a clump of cells actually. So cell suspensions are produced by, so they are actually produced by mechanical shearing of callus material in a stirred liquid medium. So what we do is we prepare a callus like you see over here. You prepare a callus and then put the callus in a culture, and due to the agitation or due to the shaking, the callus will shear off into small pieces, and that is just like a mass of 4 to 5 cells. Now, these cell suspensions generally consist of great variety of cell types, and they are less homogeneous than callus cultures, because callus is originating from a single plant tissue. So they are more homogeneous, but these cell suspension cultures, different cells are clumping together, so they might not be as homogeneous as callus cultures are. But they are still quite routinely used, mainly in those bioreactors, or we call them as biofactories. We call these cells as biofactories. And of course, you have the friable calli, undifferentiated calli, and then you transfer it to a liquid medium, and you will take it to agitation at 30 to 100 RPM, and resulting into smaller cellular aggregates, and then you start culturing them. Now, protoplast culture. Have you heard of protoplasts? I'm sure you guys must have.
Yes, sir.
What are
Yes, sir.
protoplasts? What are
Yes.
protoplasts?
Sir, cell without a plant cell without cell wall.
Okay, good. And how do we do that? Any idea?
Sir, maybe by electrical shock or mechanical shearing, we can remove.
Yes, you can do mechanical shearing actually. And then there
Sir,
are, yeah,
Cell lysis.
lysis with
Sir,
what?
you can do cell lysis.
No, cell lysis is different. We don't want to,
By
It will
using
burst open the
enzymes.
cell.
sorry?
By also using enzymes.
Yes, that's right. Yeah, that's what I was trying to come. Mechanical shearing is one thing, but we always need to ensure that it does not kill the cell. We want live intact cell wall less cells actually. So they are no longer, once the cell wall is removed, they are no longer called cells, they are called protoplast. But mechanical shearing is one way, and chemical method is another, by using enzymes, especially called lysozyme, meserozyme, all these enzymes. So this is also a routine procedure in the lab. In my lab as well as in other labs also. So this is how the protoplast, they look underneath a microscope. So we do a vacuum treatment in the presence of the enzyme solution. And this is how they look like. And if you start culturing them, you see this is how it starts looking. This is the first division that takes place. And then it starts forming a clump of protoplast like this. And the main purpose of using this is one of the main purpose of using this or doing protoplast culture is interspecies hybridization. Say when we want to hybridize two species which are not possible by sexual crossing, because they are two independent species, they are not compatible for sexual crossing. So people use these kind of protoplast fusion techniques where they fuse the protoplast from one species with the protoplast of another species, and then you culture them, they form the zygote, and then you culture them to lead to the proper plantlet growth actually. So this is one of the objectives. We also use it several aspects of gene technology. For example, we'll get onto that. But if any one of you have any idea of GFP localization or GFP, what do you call that? Not the selectable marker, the GFP localization, which is used as a, it's not used as a selectable marker. There is another term for that. Reporter gene. Okay. For using GFP as a reporter gene. Has any one of you have heard about this?
In animal culture, I have heard, sir, for that fishes and all.
Yeah. So it's good if any one of you have experienced it. Arpit, are you there?
Yes, sir.
Have you not heard of this before?
GFP, I have not heard, sir.
Okay. So GFP is used as a reporter gene. So say if I want to introduce my protein inside a cell, so I'll attach a GFP protein along with it. And GFP, we all know, is a green fluorescent protein. So it fluoresces green color under specific wavelength conditions. So in where we have done the transfection, we want to see whether our protein has got in or not. So we can't check our protein of interest, but we can check the signal of GFP. We can check whether GFP is expressing and fluorescing or not. So that will give an indication that our protein has also got in. So this is that's why we called the GFP as a reporter gene. So it's reporting the presence of our gene inside the cell. So in some cases, say if I'm using a protein, if I'm working on a protein, I want to check whether it is a nuclear bound protein or it is a cytoplasmic protein or it's a membrane protein. So I'll attach a GFP along with it. And what we do, we transfect protoplast with those fusion proteins. Am I making sense?
Yes, sir. Yes, sir.
Okay, good. Keep responding, please. I mean, if the videos are not on, at least keep responding. So what we want to see is if whether my protein of interest is getting into the nucleus or the cytoplasm or the membrane. So what I will do, I'll take a plant protoplast where I want to check its expression. I'm going to check the localization of the protein. We'll put the GFP fused to my protein construct in, and we'll allow the protoplast to express the protein. And then we will check for the GFP signal, whether it's coming from the nucleus or the cytoplasm or the membrane. So these are some of the examples of gene technology, what is mentioned over here, that this is what protoplasts are used for in nowadays modern day plant biotechnology research actually. Okay, so then the other is anther culture and meristem culture. Now, culturing anthers or isolated microspores, I mean, as I mentioned, this was done by Guha and Maheshwari, and that's why they are still considered as one of the landmark discoveries in tissue culture, that you can have culture anthers under suitable conditions, and that will lead to haploid plants through somatic embryogenesis. Now, also because they will be haploid in nature, but if you can treat them with colchicine, it is possible to produce dihaploids from those anthers. And if they survive, everything goes okay, then within a year a fertile homozygous dihaploid plant can be produced. Remember, dihaploid does not mean 2n, it means n plus n. That's why we don't call it diploid, we call it dihaploid. So it can be produced, it can produce a fertile diploid homozygous plant actually by duplicating the chromosome number using colchicine. And as mentioned earlier, this is, the anther culture is mainly used for hybrid breeding. Say if you want to generate a hybrid by crossing two plants, so you generate two haploid plants, one from one parent, another from other parent, then you just cross it, so it will form a hybrid diploid plant or dihaploid plant. Then another one is meristem culture. Here we don't do any kind of callus formation, any kind of anther or cell suspension culture. We just took the shoot meristem part, the shoot tip or axillary buds, and then we allow it to grow under aseptic conditions, which occurs in occurs without an interfering callus phase, and this results into development of a meristem culture. And the main application of meristem culture is mainly in the field of horticulture for mass propagation of clones for commercial market. So you don't do any callus. We are not leading to any kind of de-differentiation and callus formation. You just meristem culture, you develop several clones and you just plant them out. So you get, this is for commercial propagation for the same plant. If, and this is especially used in cases where the propagation of a plant is difficult. Say we transport a plant from somewhere from a very, very cold place, a very, very cold country, and we are not able to propagate it here. So you can use meristem culture as one of the techniques for propagating the plant here to take it to the next generation if they are not able to flower and if they are not able to set seeds over here. You can use the meristem culture to propagate it. All right, so this, I mean, I use the free version of Zoom, and that's roughly lasts for 40 to 45 minutes. So, and so I prepare the lectures accordingly. And all the classes will be in I try to stick to that time, because re-initiating it, then some people join, some people don't join. It gets a bit unorganized actually. So this is about to finish, so I'll end the lecture here and we'll move on from here. It is good that most of you are already aware of this, so we'll move on from here tomorrow, and then we'll move on to some other aspects of tissue culture. Friday we might not have a class because Friday I have exam duty.
Okay, sir.
Yeah, and I will post this particular video because I'm recording the lecture. So I'll post this particular video on the Google Classroom. Any doubts, feel free to put your queries in the WhatsApp group. All right.
Okay,
Okay.
sir.
Hang on. This is part of the, I need to take the, where is that? This is about